So today we will start molecular basis of inheritance. Okay, this is chapter 5 of class 12th biology NCERT. So let's start. In the previous chapter you have learned that the inheritance patterns and the genetic basis of such patterns. We have learned inheritance patterns and the genetic basis of such patterns. So at the time of Mendel, the nature of those factors means genes regulating the pattern of inheritance was not clear. So, over the next 100 years, the nature of the putative genetic material was investigated, culminating in the realization that DNA means deoxyribonucleic acid is the genetic material, at least for the majority of the organisms. So, in class 11, you have learned that the nucleic acids are the polymers of nucleotides. Now, deoxyribonucleic acid means DNA and ribonucleic acid means RNA are the two types of nucleic acids found in the living systems. So, DNA acts as the genetic material in most of the organisms. RNA, though it also acts as a genetic material in some viruses, mostly functions as a messenger. So, RNA has additional roles also, like it functions as an adapter molecule, structural, and in some other cases as a catalytic molecule. It's adapter molecule, structural molecule, catalytic molecule. It is also a messenger molecule. So in class 11 you have already learned the structures of nucleotides and the way these monomer units are linked to form the nucleic acid polymers. So in this chapter we are going to discuss the structure of DNA, its replication, the process of making of RNA from DNA means transcription, the genetic code that determines the sequences of amino acids and proteins, the process of protein synthesis means translation, and the elementary basis of their regulation. So, the determination of complete nucleotide sequence of human genome during the last decade has set in a new era of genomics. In the last section, the essentials of human genome sequencing and its consequences will also be discussed. So let's begin our discussion by first understanding the structure of the most interesting molecule in the living system, that is the DNA. In the subsequent sections, we will understand that why it is important, why it is the most abundant genetic material, and what is its relationship with RNA. So let's start with DNA. So DNA is a long polymer of deoxyribonucleotides. The length of DNA is usually defined as number of nucleotides or a pair of nucleotide referred to as base pairs present in it. This also is the characteristic of an organism. For example, a bacteriophage known as Phi 174 has 5386 nucleotides. Bacteriophage lambda has 48502 base pairs. Escherichia coli has 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. So diploid content of human DNA will have 6.6 into 10 to the power 9 base pairs. So let us discuss the structure of a long polymer. So the structure of polynucleotide chain. Let us recapitulate the chemical structure of a polynucleotide chain, DNA or RNA. So a nucleotide has three components: a nitrogenous base, a pentose sugar, ribose in case of RNA, and a deoxyribose for DNA, and a phosphate group. So there are two types of nitrogenous bases. One is purine, जिसमें कि एडेनिन एंड ग्वानिन आ जाता है, एंड देन पायरिमिडीन्स जिसमें CUT cytosine, uracil and thymine आ जाएगा. Cytosine is common for both DNA and RNA, and thymine is present in DNA. Uracil is present in DNA at the place of thymine. So a nitrogenous base is linked to the OH of 1 carbon pentose sugar through an glycosidic linkage to form a nucleoside, so such as adenosine or deoxyadenosine, guanosine or deoxyguanosine, cytidine or deoxycytidine, and uridine or deoxythymidine. When a phosphate group is linked to OH of 5 carbon of nucleoside through phosphoester linkage, a corresponding nucleotide or deoxynucleotide, depending upon the type of sugar present, is formed. So two nucleotides are linked through 3-5 phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. So a polymer thus formed has at one end a free phosphate moiety at 5' end of sugar, which is referred to as the 5' end of nucleotide chain. Similarly, at the other end of the polymer, the sugar has a free OH of 3' carbon group, which is referred to as 3' end of the polynucleotide chain. So the backbone of polynucleotide chain is formed due to sugar and phosphates, and the nitrogenous bases which are linked to sugar moiety project from the backbone. So in RNA, every nucleotide residue has an additional OH group present at the 2' position in the ribose. So also in RNA, the uracil is found at the place of thymine. That's why it is known as 5-methyl uracil. Means other name of RNA is 5-methyl uracil. Other name of thymine is 5-methyl uracil. Another chemical name for thymine. DNA acts as an acidic substance present in nucleus was first identified by Frederick Mixer in 1869. He named it as nuclein. However, due to the technical limitation in isolating such a long polymer intact, the elucidation of structure of DNA remained elusive for a very long period of time. It was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. One of the hallmarks of their proposition was base pairing between the two strands of polynucleotide chains. However, this proposition was also based on the observation of Erwin Chargaff that for a double stranded DNA, the ratios between adenine and thymine and guanine and cytosine are constant and equal ones. So the base pairing confers a very unique property to the polynucleotide chains. They are said to be complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in another strand can be predicted. Also, if each strand from a DNA, let us recall it as parental DNA, acts as a template for synthesis of a new strand, the two double-stranded DNA, let us call them as daughter DNA, thus produced, would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA became very clear. So the salient features of the double helix structure of DNA are as follows. It is made up of two polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. The two chains have anti-parallel polarity. It means if one chain has the polarity 5 dash to 3 dash, then the other will have 3 dash to 5 dash. The bases in two strands are paired through hydrogen bonds, forming base pairs. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with three bonds, hydrogen bonds. As a result, always a purine comes opposite to a pyrimidine. This generates approximately uniform distance between the two strands of the helix. So the two chains are coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer and there are roughly 10 base pairs in each turn. Consequently, the distance between a base pair in a helix is approximately 0.34 nanometer. The plane of one base pair stacks over the other in double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. So now let us compare the structure of purines and pyrimidines. Can you find out why the distance between the two polynucleotide chains in DNA remains almost constant? The proposition of a double helix structure for DNA and simplicity in explaining the genetic implication became revolutionary. Very soon, Francis Crick proposed the central dogma in a molecular biology, which states that the genetic information flows from DNA, then to RNA, to protein. But in some viruses, the flow of genetic information is in reverse direction. That is from RNA to DNA. Can you guess? Can you suggest a simple name to the process? Reverse transcription. Packaging of DNA helix. Taken the distance between two consecutive base pairs as 0.34 nanometer. If the length of DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pairs with distance between two consecutive base pairs, that is 6.6 into 10 to the power 9 base pair, into this number, it comes out to be approximately 2.2 meters. So a length that is far greater than the dimension of a typical nucleus, which is approximately 10 raised to power minus 6 meter. So how is such a long polymer packed in a cell? If the length of E. coli DNA is 31.36 millimeter, can you calculate the number of base pairs in E. coli? So यहाँ पर कैलकुलेशन मैंने दिखाया हुआ है 4 into 10 की power 6 base pairs आएगा E. coli में, which is also given here. In prokaryotes such as E. coli, though they do not have a defined nucleus, the DNA is not scattered throughout the cell. DNA, being negatively charged, is held with some proteins that have positive charges in a region termed as nucleoid. In eukaryotes, this organization is much more complex. There is a set of positively charged basic proteins called histones. A protein acquires charge depending upon the abundance of amino acids residues with charged side chains. Histones are rich in the basic amino acid residues, that is lysine and arginine. Both the amino acid residues carry positive charges in their side chains. Histones are organized to form a unit of 8 molecules called histone octamer. The negatively charged DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. A typical nucleosome contains 200 base pairs of DNA helix. Nucleosomes constitute the repeating unit of a structure of in nucleus called chromatin. Thread-like stained or colored bodies seen in nucleus. The nucleosomes in chromatin are seen as beads on string structure when viewed under electron microscope. So theoretically, how many such beads or nucleosomes do you imagine are present in a mammalian cell? The beads on string structure in chromatin is packed to form chromatin fibers that are further coiled and condensed at metaphase stage of cell division to form the chromosomes. The packaging of chromatin at higher level requires additional set of proteins that collectively are referred to as non-histone chromosomal proteins. In a typical nucleus, some region of the chromatin are loosely packed and stains light and are referred to as euchromatin. The chromatin that is more densely packed and stains dark are known as heterochromatin. Euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is inactive.